Hello everybody. The Biochemical Society and Portland Press are really pleased to welcome you to this webinar. It's part of our Biochemistry Focus webinar series. Topics in this series include different research areas in the molecular biosciences, as well as some practical sessions to support your career development. And as you probably know, each webinar gives you the opportunity to ask questions via text at the end of the webinar. And we also welcome suggestions for future topics and speakers. There's further information on the website. So my name's Claire Shovlin. I'm Professor of Practice in Clinical and Molecular Medicine at Imperial College in London. And I'm delighted to be chairing today's webinar. It's titled Discovering the Potential of Epitranscriptomic Ribosomal RNA Fingerprinting. And our invited speaker, we're so lucky, he's going to be discussing the findings from his latest paper, which employed direct RNA sequencing to chart human and mouse rRNA epitranscriptomes across tissues in development and in disease. So before I hand over to our speaker, I'd just like to mention that the questions will be asked at the end of the webinar, but please do send in your questions during the talks. If you have a question, please type it in the question box as shown in the image on the screen, which is just there. And we'll try to answer as many of these as possible at the end. Now, our invited speaker today is Ivan Milenkovic from the Center for Genomic Regulation in Spain. Ivan obtained his PhD here in Spain under the supervision of Dr. Eva Novoa, and the focus of Ivan's research is ribosome heterogeneity at both ribosomal protein and rRNA levels. Ivan's currently doing a bridging postdoc in the Novoa lab, where he's working on developing new therapeutic strategies to tackle heart-specific ribosomopathies. So I'm pleased to hand over to Ivan, and Ivan, I can see your slides, so that's super. So over to you.
Splendid. Thanks a lot, Claire, for the very nice introduction. Thanks to the organizers for inviting me, and to everybody who's in the audience today. So in today's webinar, I will be talking about my PhD project, which was focused on ribosomal RNA heterogeneity at the level of RNA modifications, and how we came up with a new method that we decided to call epitranscriptomic RNA fingerprinting, that allowed us to chart tissue of origin and tumor-specific signatures. So basically, just to set the stage a bit, I'd like to just give a brief introduction on ribosomal heterogeneity and ribosome specialization. So I'd say that through history, in biological sciences, ribosomes were mostly perceived as static entities. So they would be there to translate mRNAs, to synthesize proteins, but nobody, not nobody, but like most people were not giving much attention to control, translational control at the level of the ribosome. So it was there to translate the transcript and to produce the protein, but we thought of them as static and uniform entities. However, this has been challenged over the years with different ideas, different hypotheses, and different projects that came up with some of the first results pointing towards ribosome heterogeneity. So you have on the left here, you have a timeline that shows how everything started with the one gene, one ribosome, one protein hypothesis that was proposed by a long time ago, but it was not really perceived so well. And then over the years, we had more and more results that were kind of pointing towards the direction that ribosomes are more heterogeneous than we used to think. So basically, if you would take a ribosome from a skin cell and from a brain cell, you would expect them to be the same, but this is not always true. And these differences, so this ribosome heterogeneity, can occur at different levels. For example, at the level of ribosomal protein stoichiometry, so you can have different ribosomal proteins being incorporated into the ribosome. You can have different ribosomal protein paralogs being incorporated into ribosome, RP modifications, associated factors, then the localization, the subcellular localization of the ribosomes. But the one that I was most interested in, especially in the second half of my PhD, were ribosomal RNA modifications. And this is what I'm going to be talking about today. In the past couple of years, so let's say in the past 10 or even more in the past 5 years, a few papers have come out that have shown that these differences at a level of ribosomal RNA modification are functionally relevant for ribosome specialization. So they were discovered some time ago, but it wasn't really clear whether they really had a functionally relevant role in the cell. It turns out that they are dynamic in development, so they're going to be different ribosomal RNA modification patterns in embryonic and adult tissues, for example. This was done in different species. It's important. There was a very nice paper from 2 years ago that showed that specific ribosomal RNA modifications are important for cell fate commitment. So from stem cells to different cell lineages, that the ribosomal RNA modifications can influence the translation of CG-rich transcripts. So it has a very direct functional role on determining how transcripts are translated. And then that there are different cancer-specific ribosomal RNA profiles when compared to healthy tissues. Now I'm a biochemist by training, so I'm very fond of chemical formulas, and I thought it would be nice to have an overview of the different ribosomal RNA modifications that have been annotated and described so far. So these are the ones that are present in human and in mouse. They're more or less the same. As you can see here, for each of the bases, there are different modifications, and then each base can have 2' methylations on the ribose. Sorry, each nucleotide can have a 2' methylation on the ribose. So there can be AM, which would be ribose methylated A, CM, UM, and GM, and so forth. So these are the ones that have been annotated so far, and I would say that ribosomal RNA modifications are really well studied. So we have a very good idea of where they are in the ribosome. They've been charted in mouse, in human, in yeast, in E. coli, etc. So these annotations exist, and they work very well. So if anybody's interested in looking at ribosomal RNA modifications, I think there's a lot of resources out there that you can start. Now, I think that it's very important also to point out that ribosomal RNA is heavily modified. So if you took a look at the either 18S or 28S, this is in mouse, for example, you would see that there's a lot of ribosomal RNA modifications throughout both of the molecules, the long ribosomal RNA molecules. And then not only this, but you will see that there is a specific type of modification that's more abundant than other ones. So in this scheme here, you will see there's a lot of red and black dots, and they represent either NMs, which are 2' methylations, or pseudouridines. So these two modification types represent around 95% of all of the modifications found in ribosomal RNA. And there have been efforts in the past couple of years to chart the dynamics of ribosomal RNA modifications. However, these efforts were always relying, or almost always relying, on methods that can only chart one type of modification at a time. So one of the most well-known ones is RiboMet-seq, which is an Illumina-based method that basically captures the 2' methyls, but it's blind to all the other modification types. On the other hand, one can use pseudo-seq that can only capture the red dots, the pseudouridines, but then you don't see the other modification types. And as you can see, there's many other ones. So what we decided to do in our lab was to try and use direct RNA nanopore sequencing, which I will get into in the following slides, that at least in theory can provide you with information about all the modification types found in ribosomal RNA. Of course, in practice, this is a little bit more difficult. It's not really that straightforward. But at least when we were starting the project, we thought, okay, this is the capacity of nanopore sequencing. Whether we're going to reach the full capacity, we weren't sure, but we wanted to give it a go. Now, maybe not everybody is familiar with nanopore sequencing, so I would just briefly go over how it works. This is a figure from a recent review that a colleague of mine from our lab published in Cell. So I would also encourage you to give it a read if you're interested in technologies based on nanopore sequencing. Basically, the way it works is that you have your nucleic acid, which can be either RNA or DNA. So you extract the nucleic acid from any material. It can be cells, it can be tissues, it can also be in vitro transcribed RNA or other types of samples. And basically what you do is that you prepare a library and you take the exact same molecules that were in the case of RNA sequencing, You take the exact same molecules that were found in the cell, and you make them go through these nanopores, these protein nanopores. And this is basically what's revolutionary about nanopore sequencing, because most of the other sequencing methods always relied on a cDNA sequencing step. So you would have to synthesize the cDNA of an RNA molecule, and this is what you would be sequencing. When it comes to nanopore, you're actually sequencing the exact RNA molecule that was found in the cell in the first place. It can, as I said, it can be both DNA and RNA. I will focus mostly on RNA here because this is what our lab is mostly using. So you're going to have your RNA going through the pore. So this is a protein pore in a membrane. And as the RNA is going through the pore, each of the nucleotides found in the pore is going to affect the electrical signal that the system is recording. So in the sequencer, there's going to be a detection method, let's say, that relies on the electrical signal. And every time you have a nucleotide going to the pore, this signal is going to be slightly different. Now, not only the signal differs from nucleotide to nucleotide, but it also differs from an unmodified to a modified nucleotide. And this was very useful for us because we thought, okay, we can use this method then to chart RNA modifications, not only ribosomal RNA, this is what I was mostly working on, but we can also look at other RNA types. And there's 3 different ways you can do this. You can look at the current feature, so you can look at the electrical current, and you will see that in the unmodified base, you're going to have one type of current intensity profile here, and in the modified, here in red, it's going to be different. The other way is to take whatever current intensity profile you get, you base call. So this is basically transforming the current intensity to bases, to the sequence. And with this sequence, you basically map to a reference. And what you would often get is an error, is a base calling error that's found in the place where the RNA is modified. So due to the modification being present in the base itself, the base calling algorithm would misread the current intensity, and it would mislabel it as a different base. So you could use these errors as a proxy of modifications. And there's another way of using modification of your base callers. So basically, to already at the level of base calling, you train the algorithm to recognize modified bases. Also, we have a new paper that came out just a few weeks ago that was done by my colleagues that focuses on developing a new base caller for M6A. So you would have an M6A base caller that differentiates non-modified A from M6As. And there is quite a few downstream analyses that you can do from here. I will not go through all of them, but I will just tell you that the one way that I decided to use for this paper and for this project is this second one here. So alignment feature. I decided to use the base calling errors. And this is the way it works. So imagine that in the RNA, you have a modified U. And what can happen are 3 different things. One is that this U is read as a C. So in the read, you're going to have a mismatch. There's going to be a wrong letter assigned to this base in the read. What can happen is that the base calling algorithm will not know what to do with this modified base. So it will just not introduce anything there. So this will be read as a deletion, or since it's unsure what sense to make out of the signal, it can insert additional bases. So this can be an insertion. Of course, these errors depend on the sequence context, they depend on the modification type, but basically what we realized one can do is take these 3 different error types and sum them all together in a single value called sum errors. So basically the sum error is going to be a proxy of the modification status of a given base. And this is what I was mostly using to look at ribosomal RNA modifications. Now, during my PhD, I was also working on ribosomal proteins. I was working with mice. I was interested in ribosomes in mouse hearts. And while we were also working on proteins, we decided to also look at ribosomal RNA. So I decided to use more or less the same model system. So this would be mouse tissues. And we decided to take 2 different dimensions into account. So we were interested in ribosomal RNA modification dynamics across different tissues. So to see if they are different in a brain, in a heart, in liver, and so on. And during development. So we had embryonic, newborn, and adult stages. So to sample all of these tissues from different mice. So this was done in 2 biological replicates. And from all of these samples, I would extract total RNA, I would prepare the libraries, I would sequence them using direct RNA nanopore sequencing, and then I would calculate the summed errors for each base, for each RNA, for each sample. And now once I had these data frames, so this was a lot of data, I could then compute the, let's say, median of summed errors for each of the samples. Like I would take all the samples, compute the median, and then compare each of the samples to the median to see which sites are less or more modified when compared to the median. So for example, here, this is just an example, but you will see that some sites are outliers on this side of the diagonal, so they are increased, and some are decreased. So with this and doing these one-to-one comparisons for all the samples to the median, I could then come up with a list of dynamic sites across these samples that were taken into account in the first place. And this is what the results look like. So this is a heat map of ribosomal RNA modification dynamics of these outliers. So these are the sites that were identified as being dynamic, either across tissues or in development. And you will see that there are some interesting patterns here, that some are increased in embryo, some are decreased in adults, some are decreased in embryo. And then the ones that we thought were the most interested from the very beginning are these ones here that seem to be increased in adult brain. So it seems that when you look at these tissues and these developmental stages, the ones that pop up the most are adult brain ones. So for some reason, there are some ribosomal RNA modifications which are highly specific for adult brains. We didn't really know this before, and we still don't know why this is the case, but for sure it's happening. And here I would like to show you some of the examples of dynamic ribosomal RNA modification sites. Just a brief introduction on how to interpret these plots or these visualizations of reads. This is basically an IGV snapshot where you will have embryonic, newborn, and adult tissues in green, red, and blue. And every single rectangle here corresponds to one base, and you have the reference bases in the bottom. So every time the base in the read corresponds to the reference, it's either not going to be colored or it's going to be colored with these pastel colors that I selected, so green, red, and blue. But whenever there's a mismatch, so whenever U is read as a C, for example, you will see these bright colors here. So what this means is that in let's say half of the reads, this U, so it says T here because the nomenclature usually is DNA centric, but in RNA these are uridines, it's going to be read as a C. And we know that this often happens with pseudouridines, that if you have a pseudouridine in this position here, it's often going to have this mismatch pattern, U to C. And you will see that across all the tissues, this is very consistent. It doesn't really change, except for adult testes. So for some reason, in adult testes, this specific base is not modified anymore, or very lowly modified when compared to all the other ones. And there's other examples, such as the 18S-U355, which is a UM, so ribose methylation of this base, which goes from less modified in embryo to more modified in newborn and adult. Then there's some other ones which seem to be adult specific. This is one of the ones that I was referring to earlier, that's an adult brain specific one. And this one here was more of a control. So this is a very well-known hypermodification. It's called M1-ACP3 pseudouridine. And you will see that it's fairly constant across all the tissues. Now, there's one more disclaimer I have to make here. We use the same samples to also do a cDNA run, because we had to make sure that these mismatches were coming from modification and not SNPs. So if the difference had been at the level of the sequence, we would see it in cDNA as well. But as you can see in these 4 examples, there's no mismatches in the cDNA. So it's coming from the modifications and not from SNPs. However, in the case of M1-ACP3 pseudouridine, we do know, it's well documented, that this modification can impair Watson-Crick base pairing. So you are expecting to see a mismatch also in the cDNA. And this is what you see here. So basically, this is just for you to get an idea of what these dynamic modifications look like in terms of reads themselves. Now, in the bottom, you will see that some of these modification sites have annotated modifications. So we know that there's a pseudouridine here. We know that there's a 2' methylation of the uridine here. But in the case of these 2 here, these were novel putative modifications that we discovered while we were doing these experiments. So we didn't know if they were modifications at all to start with, and then if they were, what type of modifications there were. For example, here in the example of U1359, this really looks like a pseudouridine because it's a U that has a very clear mismatch U to C, and we thought this could be a pseudouridine. So what we decided to do is to use an orthogonal method called nano-CMC-seq, in which you take your RNA, you fragment it, and you treat it with a chemical called CMC. And this CMC will react with pseudouridines. So it will form a novel moiety on the pseudouridine. So when you do a cDNA synthesis on this template that has these CMC labeled pseudouridines, the reverse transcriptase will not be able to continue the reaction past the modified pseudouridine. So in this way, you could identify where in your transcript you have pseudouridines. This was also published by a colleague of mine from our lab some years ago. And we could see that some of these sites, the one I was showing earlier was the 1359, are indeed pseudouridines, and they do indeed follow more or less the same trend of being enriched in adult brain. As you can see, 1359 is, you could identify it in adult brain, but in adult liver, it was not really modified at all. So we were happy to see that not only can you chart the ribosome RNA modification dynamics using nanopore, but you can also discover new sites that were not identified before. We were also interested in what happens in vitro. So if we can study these things in vivo, which are of course very good systems to study certain things, but then also complex systems for other reasons, because not always can you rely on in vivo systems for all the experiments you want to do. We were interested if we can also see something similar if we differentiate stem cells to NPCs, so neuron precursor cells and neurons, kind of to mimic what happens during development from an embryonic brain to an adult brain. And we could see that for some specific cases, for example, the uridine 1315, which goes from less modified to more modified during in vivo development, we see something very similar in vitro. So from stem cells where it's not really modified to very modified in neurons. So we were happy to see that some of these effects you can replicate in vitro, but then not all of them. So not all the results that you can get in vitro, for example, from a differentiation system, should be directly applied to in vivo systems. So for example, this modification here seemed to be in vivo specific. We didn't capture it at all in vitro samples, then vice versa for this one here. And then for this modification here that seemed to be enriched in the adult brain, we didn't capture it at all in any of the cells. However, now there's other examples of modifications. I was also referring to this modification before when I was only looking at the in vivo data. So now we compiled all the samples here, so cells top and then all the in vivo data in the bottom of the U355. It goes from less modified to more modified, and we thought it seems that this modification is inversely correlated with the, let's say, differentiation status of the cell or tissue. And if you look at the summed error values, so it goes from less to more from stem cells to neurons, and it goes from less to more from embryonic to adult tissues. And we thought, okay, this is interesting. Now, the way ribosomal RNA modifications work when it comes to pseudouridines and ribose methylations is that they are deposited by specific SNO RNAs. In the case of the U355 modification, the corresponding SNO RNA is called SNORD90, and it wasn't completely clear if this is really the SNO RNA that's depositing this modification, I mean, guiding the enzyme that deposits the modification. So we were also interested in checking this because based on bioinformatics, it should be corresponding, so there is complementarity in the sequence, but this hasn't been shown before. So we're interested in generating SNORD90 knockout stem cells using CRISPR, and then differentiating these stem cells to neurons, and then comparing wild-type neurons with SNORD90 knockout neurons. And we could indeed see that the modification is lost in the SNORD90 neurons. So this is for sure the SNO-RNA that's responsible for modifying this exact spot. We then looked at the morphology of these neurons, but we didn't really see any differences. So SNORD90 knockout neurons are fine. if you just look at the morphology. And a colleague of mine, Mia, from the lab, is now working on characterizing these cells and figuring out what exactly is the role of this 18S U355 modification that seems to be very robustly dynamically regulated in terms of being less modified in more stem-like systems, so embryonic tissues or stem cells, and more modified in more differentiated cells and more adult tissues. Now, when we were looking at all these different modifications, we were starting by looking at the differences. So we were like, okay, let's see, what's enriched in the adult brain or what's depleted in the embryo liver. But then we realized that there are some patterns here that might be actually important and informative. So we did a PCA of these dynamic ribosomal RNA modification sites to see if based on them we can cluster tissues together. And you will see that here adults, heart, brain, liver, and testes cluster very nicely together. So this means that only based on ribosomal RNA modification profiles that we decided to call fingerprints because they seem to be informative enough to tell tissues apart, you can make a PCA where different replicates of the same tissue cluster together. And the reason is basically shown here in snapshots of a few select sites. So for example, in the 18S407, there's going to be, maybe it's not so easily seen by eye, but in liver, it's more modified than other tissues. In the case of 18S1178, this is the one I was talking about before, which is depleted in adult testes. This is the one that's adult brain specific, and so on and so forth. So basically, based on these patterns, or as we decided to call them, epitranscriptomic fingerprints, you can tell tissues apart. And with this realization, we thought, okay, maybe we can actually train a model. We can take a model and try to train it to tell tissues apart, to infer what type of tissues you have only based on ribosomal RNA modification patterns. So the idea would be you take ribosomal RNA, you sequence it, you analyze it, you train a model, and then you try to predict tissues based on these fingerprints. The way we did this was to take the BAM files, which are basically the mapping files of ribosomal RNA, where you have the sequence mapped to the reference, and to computationally split them in 12 pseudo-replicates. So just randomly taking 10%, 20%, 30% of the reads, then take these pseudo-replicates and run the pipeline on them. So EpiNano is the software we use to get the summed error values for each site for each sample. And then we used random forest classification to predict the tissue type. So this was done on 3 biological replicates. The first one was used for training, the second for testing, and the third for validation. And this worked very well. Not only that, but since ribosomal RNA is highly abundant and you can get a lot of reads, we also thought, okay, what if we reduce the number of reads that we are using in this experiment? So for example, if you take a Milenkovic, which is the standard sequencing device for nanopore, nowadays, if you do a ribosomal RNA run, you could get anywhere between 1 and 2.5 or 3, even more than 3 million reads. So if you think about it, you could put whatever, 10, 50, 100 samples, given that you have a tool to multiplex, because this is not really something that's commercially available. It's also something that's being developed by our lab. So if you can multiplex samples, you could in theory put 100 samples in one sequencing flow cell and still get a lot of reads per sample. So we were interested in seeing what's the minimum amount of reads that we need to be able to predict tissues properly. And you will see that if you go to as low as 100 reads, you will still get a very good, so this is a rock curve, you will still get a very good performance of the classifier, which means that we decided to go with, let's say, 400 reads, which is already a very low number if you're considering that you can have up to 2 million in one flow cell. One flow cell costs between 500 and 1,000 euros, I'd say, depending on how many you order in one batch. So this basically brings you to a very low cost per sample if you're interested in classifying tissues, for example. And this was also extraordinary. Like, we were very excited to see this, because this means that you could, in theory, also go down to the single cell level, because in single cells, it's very hard to get enough material, whatever this material might be. It could be proteins, mRNAs. With mRNAs, it's particularly difficult because they're not so abundant. But ribosomal RNAs, which are the most abundant in terms of RNA content in the cell, this could then mean that you could, in theory, do identification of cells based on ribosomal RNA. So we're very excited to see this. This doesn't only work with telling tissues apart, so identifying tissue types, but if you take one tissue type, for example, brain, of different developmental stages, so embryonic, newborn, and adult, you will see that the PCA also looks quite good, that the different stages cluster together. And then when you run the same pipeline with the random forest classifier, in each of the cases, it would predict all the 3 stages correctly. So this means that you can also use this to infer the stage of a given tissue, not only the tissue type. We were then interested in seeing whether this also works in vitro, so with our cells that we differentiated from stem cells to neurons. And it was very similar, so it correctly classified all the cells that we looked at, so the 12 pseudoreplicates of stem cells, of NPCs, and neurons. So this means that epitranscriptomic fingerprinting can be used both for cell type detection, for tissue type detection, and for developmental stage detection as well. While I was looking at these results, I was also wondering how it was kind of exciting to see that the sequence of ribosomal RNA has been used for detecting, for example, bacteria for years, for decades. It's like a very optimized method that's been used throughout the world for prokaryote classification. And it turns out that not only the sequence of the ribosomal RNA is super informative, but also the epitranscriptome. So whatever is found on top of the sequence could be quite informative as well. And for me, it was important to wrap my mind around this because in most cases, ribosomal RNA reads are considered rubbish. So it's what most, like when you're interested in transcriptomic studies, for example, when you're looking at RNA expressions, if you ever have any ribosomal RNA reads, one of the steps in the analysis is going to be filtering those out. So the current consensus in the, say, transcriptomic field of analyzing RNA-seq and related data sets is that ribosomal RNA is far from informative. And we were excited enough to see that this is not really always the case, and that maybe ribosomal RNA modifications can be quite an important source of information. So based on all this, we decided to maybe go further and to take a look at clinically relevant samples. So we ordered commercially available matched cancer and healthy tissues of colon, liver, lung, and testes. This means that from one same patient, you will have a matched pair of a cancer tissue and adjacent normal tissue. The idea was then we could then compare these pairs to see whether ribosomal RNA profiles were different between the cancer and the normal. And this was the case in some of the tissues, but not all of them. So for example, here on the right, you will see that in the case of lung and testes, there's a lot of outliers. And that the ribosomal RNA profile is evidently different between these two samples. However, in the case of colon and liver, this was not so clear. So what we decided to do here is to take the top 20 most dynamic ribosomal RNA sites across these samples, and then to try and see if we can plot a PCA that's going to be clustering normal samples on one side and cancer samples on the other side. So this PCA was not the most beautiful ever. So you will see that there's some overlap here, that liver is overlapping with, the cancer liver is overlapping with normal colon. So you couldn't really tell these apart, but it was the first step at seeing that, there are some differences in terms of ribosomal RNA modification patterns between cancer and normal. And it also helped us decide which tissues we would maybe want to focus more on in the future. It's important to say that, as I was saying in the beginning, we had to do, when we're looking at ribosomal RNA modifications using nanopore, we had to make sure that those were not coming from SNPs. So when working with cancer samples, I think this is even more important. So what we did here was to take ribosomal RNA that was sequenced using direct RNA nanopore sequencing. And then we synthesized cDNA using the same samples. And we saw that all these mismatches that we see are not present in the cDNA tracks, meaning that whatever differences we see at the level of mismatches are not coming from SNPs, so they're coming from really different modification patterns. And already here by eye, you'll be able to see that some modifications are found in some samples and not in others. But since we only had one replicate per tissue here and only 4 tissues, we thought, okay, this is just like a pilot experiment, but we kind of have to go further and deeper with the number of samples and to be more focused on one tissue type. So the next step was to get our hands on as many clinically relevant samples as we could. So we started a collaboration with Karsten Müller-Tidov from the Heidelberg University Hospital, who has access to clinical samples. And the idea would be to take these matched pairs of normal and cancer samples to do RNA extraction, to do the sequencing, and then to use one part of the data set for training and the other one for testing whether a random forest classifier can predict whether a tissue is normal or cancer. And this is what this looked like. So we had 40 samples, so basically 20 matched pairs. And just by doing the PCA to start with, you could really nicely see, and this was like one of the most exciting moments of my PhD, is to see that just by looking at the most dynamic sites, you could easily separate the normal from the cancer sample. So this means that with something very simple, so using ribosomal RNA modifications coming from a nanopore sequencing experiment, which can be done in a day basically, you could fairly accurately tell apart whether a tissue is normal or cancer. And these are the snapshots of some of the select bases that were modified differentially between the two groups. Now, it's nice to see this by eye, but we also wanted to quantify this. So what we did was to take the first flow cell, in which we sequenced the first 20 samples, so the first 10 pairs, and then we trained the classifier based on the results from this first flow cell, which we then tested on the second flow cell. So even if there were some batch effects between the flow cell 1 and flow cell 2, which were like sequenced on different days and so on, this would be taken into account. So we would see if these findings are robust enough to train a classifier that would then correctly predict whether something is cancer or normal. And lo and behold, when we tested the classifier on the results from the second flow cell, it accurately classified all 20 samples. And not only this, but we were also interested in looking at the minimum amount of reads that we need for this classifier to work accurately. And again, with as few as 250 reads, this was working very, very well. So this was published there recently in Mall Cell. So if you're interested, feel free to take a look at the paper, and you'll see more information about the whole project over there. What I think is very important here is to say that with having an approach that can rely on as few as 250 reads to classify between tumor and normal, this really allows us to scale things up enormously. So in one flow cell, as I was saying, that can cost between 500 and 1,000 euros, you can in theory put 100, 200, 500 samples to do some pre-screenings, to do all sorts of things, and this would be really, really low costs per sample, which I think is very interesting for the future diagnostic approaches in the clinic. And with this, I'd just like to go over a few conclusions. So I hope I convinced you that ribosomal RNA modifications are dynamic across tissues in development and in differentiation, that direct RNA sequencing detects annotated and previously unannotated ribosomal RNA modifications. So thanks to these efforts of ours, we now know of 4 more modification sites in mouse and human that were not annotated before. In vivo and in vitro ribosomal RNA modification patterns are different. So if you have some results from an in vitro system, these do not necessarily apply to all the in vivo systems. What we termed epitranscriptomic fingerprinting, so using ribosomal RNA modification patterns to tell things apart, can predict tissues, stages, cell types, and cancer. and it's accurate with SQS 500 reads, making it a promising tool for diagnostics and future adaptation for the single cell level. This is something I'm very excited about, and I think it's going to happen sometime soon. We also have some work along those lines in our lab, and I think that since for now we don't have any RNA-based sequencing methods for single cells, this might be one of the first ones. And with this, I'd like to thank my lab, especially Eva for supervision, all the alumni from our lab, the funding bodies, and the collaborators. And here you have my email as well. If anybody is interested in getting in touch and discussing any of these results or anything else, please feel free. And I'm happy to take questions.
So thank you so much, Ivan. That was a superb talk, and many congratulations. Really exciting. We've already got lots of questions coming through from the panel. So please, if you've got a question, do type it into the question box as we can then go through them. So I think the first question is from Walid Albihal, who says, thank you for a really great and interesting talk. And the question is, with the focus on a single rRNA modification, wouldn't it be simpler to use mass spectrometry to detect? And I think that's a really important methodological question. So I wondered if you'd be able to address that for us.
Right. I think mass spectrometry is a very important approach that is by far more important than nanopore in a number of, let's say, applications. But however, one of the downsides is that you don't get the sequence information. When you use mass spectrometry, if you're interested in let's say, quantities of modification type, it's very precise, it's very sensitive, and so on and so forth, but you don't get the sequence information. Also, what nanopore is very cool in terms of novel applications is that you can look at two different modifications, even two different modification types, in single reads. So you can see if two modifications are co-occurring, if when you have one modification, the other one is not there anymore. So even though nanopore has its downsides, I think there's quite a few things where nanopore outperforms mass spec. And I think the two should be used together. So it shouldn't be a competition, but I think one should design their experiments in a way that they make the most out of all the possible methods we have at hand.
That's really, really helpful. Thanks. I think that'll help guide a number of people. And a slightly related question from me, actually. Obviously, you alluded to the fact that ribosomal RNAs are so much more prevalent in the cell than the other RNA species. We might be interested to say tRNAs or mRNAs. Do you think that the same holds for the other species where it will be harder to detect them just simply because of the numeric number of RNAs within the cell?
So if I got your question right, you're asking if other RNA species are going to be as easy to detect with nanopore sequencing, yep. Right? So no, if you take a total RNA extract and you don't pre-treat it in any way, if you prepare the library and you sequence, you're going to get 90 to 95% ribosomal RNA reads. So there have to be some steps prior to library prep where you will select for an RNA species. So it can be whatever, ribosomal RNA depletion. So you ribodeplete, and then you're left with mostly mRNAs, long non-coding RNAs. You can select for short RNA, so you can be sequencing tRNAs and snRNAs. So in our lab, we also developed an approach called nano tRNA sequencing that uses nanopore sequencing for tRNAs. It's not as easy as what I did here because really my life was very simple. I would extract RNA from cells or tissues and I would sequence that. If you're interested in RNA in other RNA species, it's feasible, but it's not as easy.
Yeah, thank you. So we have a question from Sudeepa Das. First of all, saying wonderful session. Does the modification seem to be consistent in heart tissue from embryonic to adult stage, going by the error bars? And I wondered if that was that was the really asking for the clarification on your error bars in the consistency of your findings.
I guess it was this one here, because I'm not sure if other plots had error bars. So I have to say what happened here was that I had two biological replicates, and in the case of the embryo heart, one of them had lower coverage than the other one. So the error bar in the embryonic heart was this high because of the difference in the coverage between two replicates. But I think that in general, what we saw in the heart, so levels going from, in case of 18S355, levels going from less to more when you go from embryonic to adult tissues. This was true for all the tissues we tested, including the heart. Now, for the other modification types, I have to say the heart didn't really have any very specific modifications, only that we saw in the heart, like we saw for brain, for example. So if you go back here, there were cases like this that we saw modifications only in adult brain. So the heart, when compared to all these other tissues, it didn't have a very, very specific, let's say, different ribosomal RNA pattern when compared to all the other tissues.
Right. Thank you. Thank you. And now a question about obviously you showed us the very many different types of RNA modifications that are present, and you picked the most dynamically changing ones as part of the signatures. Karina Aldale has asked if there's any interaction between the different modification types. Are there any patterns that you can see? Is a good question.
So I have to say it's a very good question. I have to say we haven't looked at it yet. We had the idea of looking at that through some other data sets, but it's true that even in this data set, we already have all the data we need to look at different co-occurrences. There was just so much one could do, and I think this could be a nice side project for us to take a look at in the future. But yeah, my answer is that it would be very interesting to see if there's some co-occurrence of different modifications, but we just haven't looked at it yet.
Yeah. Okay, thank you. And the probably a slightly similar question about this is one from Leonard Liam Van Dorp, asking if there are particular modifications that showed stronger predictive power for the cancer diagnoses.
Yeah, so there were a few. There was one that was very interesting. It was the 18S296. So this is an annotated pseudouridine, the one here, and it was the only one that was more present in cancer tissues when compared to normal. So all the other ones were mostly less modified in cancer when compared to their normal counterparts. However, this one was the one that was kind of sticking out as an exception. So I would say that this is a very interesting one. There was another paper that came out a few years ago that used one of these pseudouridine specific methods to only look at pseudouridines in cancer. And I remember that they could not, for some reason, look at this specific modification because their method was too aggressive for this part of the ribosomal RNA. So basically, they wouldn't get coverage in this part of the ribosomal RNA. So they said, we have no information about 18S296. And I was very excited to see that one of the most important ones in our hands was this modification here.
That's all right. That's interesting. While we're staying with the cancer, the modifications that you're describing, obviously, the developmental and the tissue specificity appears to be regulated. Do you think this is a part of the causality of cancer, or is it reflecting what's happening as a consequence of trying to improve proliferative rates in a non-causal method?
This is a very good question, and this is something I would be super interested in finding out. So we haven't done much along those lines so far. Once we realized that these fingerprints can be used as a source of information, we kind of focused on that. But the biology behind it is very interesting. We have some efforts in the lab now that are trying to tackle this, but I mean, it will be a long project. It would take a lot of effort both in the wet lab and bioinformatically. I would say that my educated guess, but this is just a guess, right? Would be that there's going to be both cases. There's going to be some modifications that are going to be, let's say, if cells are proliferating more quickly, there's going to be some modifications that are going to be less present just because there's, you know, a lot of cells proliferating and the modification machinery is not keeping up with the proliferation. However, I do think that based on some of the results that we're seeing before, so I was referring to this paper in the beginning, this CM174 modification that's important in cancer that was shown to be, so it's downregulated in cancer. This was a paper from 2021. They saw that this affects the translation of some specific transcripts that might give an evolutionary advantage to cancer cells. So I would say that we identified a number of dynamic sites between normal and cancer, and some of them could as well be just as different between proliferative and non-proliferative non-cancer cells, and some of them might be actually important for cancer biology.
Yeah, really interesting. And we have a question now from Swali Swero, also on probably the cancer, but maybe broader. Again, thanking you for the really interesting talk. Could rRNA fingerprinting be applied in precision medicine to tailor treatments based on individual epitranscriptomic profiles?
This would be my dream. This is like one of the things that I really look forward to. I think it's still far, but I think the idea is the following. So imagine you have a ribosomal RNA sequence that ends up, so the ribosomal RNA molecule that ends up in the ribosome. And it has more or less of certain modifications in the cancer. And these patterns can then be used to tailor make drugs that are going to be specific for those ribosomes. So imagine a drug that would target cancer ribosomes, so an oncoribosome, but not the other healthy ribosomes. So this would be something very interesting, very new, because so far ribosomes were not perceived as druggable targets because you can't really differentiate ribosomes between cancer cells and normal cells. But it turns out that maybe you can. And what's even more exciting, if I can just add a few more sentences here, is that now people are developing methods where during surgery, so if a patient goes into surgery and they are under anesthesia and the surgeon takes a piece of so they excise the tumor and they take a piece of the tissue, they give it to the lab, and in a few hours they do nanopore sequencing on this tissue. So they only did this with DNA so far to get a methylation profile of or a mutation profile of some oncogenes of interest. Based on the results of the sequencing that can be really streamlined and done very fast, so in the matter of a few hours, so the patient is still in the surgery room, the surgeon can get the information and decide which type of treatment to use before patching up the patient. So I thought this could also be something interesting for epitranscriptome fingerprinting. So if you have a patient with a tumor, with a specific tumor, you can then get some information about the ribosomal RNA profile of the tumor ribosomes, and then decide what type of therapy you would use there. Of course, this is years away, and I'm sure that we are nowhere near developing these drugs, but who knows? I think there's some promise lying there.
Yes. Well, thank you so much for these. There are a few other questions just to run through. You've probably only got time for you to select one of these. One is referring to the other types of RNAs and whether lincRNAs, SNO-RNAs, and the others have the modifications, how these are written, read, and erased. And another one is about dynamic questions during, say, stress responses. So I don't know if you'd like to pick up on any of those in the last minute or so.
Sure. So the first one, so regarding the first one, I have to say I'm not an expert in terms of other RNA species. I do know that they are modified, SNO-RNAs especially. And I think in terms of readers, erasers, and writers, we know a lot more about mRNA. So we know there's specific enzymes that place modifications, some that read them and some that remove them. In terms of ribosomal RNA, we only know which enzymes place them. So we don't know about erasers, for example. And I would say that it's similar for SNO-RNAs, and I would guess lincRNAs as well. But as I said, I'm not an expert. I'm quite sure that they are modified as well. We were looking at some. So what you get extra when you do a total RNA sequencing run, even though 90% of your reads are ribosomal RNA, you can sometimes get some coverage on SNO-RNA. So sometimes you would take a look at these remaining 10% of the reads, and you would see some modifications in those as well. And then we had some collaborators who were more interested in SNOR RNAs, for example, so we were sharing our results with them. But yeah, they are definitely modified, and I'm not 100% sure if they are also read by specific enzymes and if those modifications are erased. I'm happy to answer the other question as well. If you can just remind me what it was.
It was about the dynamic changes in terms of acute responses to cellular signals and
right.
perhaps stresses.
Right. This is also very interesting. We have done experiments in yeast where we wanted to see if there are dynamic responses in like heat shock or oxidative stress in terms of ribosomal RNA, and we didn't find any differences. But we did find differences in SNOR RNAs, for example. So it seems that ribosomal RNA modification profiles are not the most responsive, at least in the short term, to external stimuli. But this was only one system that we tested, so it was only yeast. Maybe in mammalian cells it'd be different. And we are also interested in looking exactly at this. I'm doing some experiments now with some cells in culture to check what happens with ribosomal modification.
So that's lovely answers to all those really great questions. So I think it's probably time now for us to wrap up. So I'd first of all just like to thank everyone for attending, and particularly Ivan, our wonderful speaker. You can continue this conversation online by following at BioChemSoc on Blue Sky and X. We also welcome suggestions for future topics in these webinars and speakers to feature. So if you have an idea for 2025, please do submit a proposal, and you can find more information about the webinars at our website, www.biochemistry.org. Now, you'll find all of the upcoming webinars also listed on the website as well. And if you've missed any of the 90 or so webinars that we've already ran as part of the series, or if you'd like to watch them again, just visit the website or the YouTube channel, and there should be the details will be shown on the screen. We expect the recording from today's webinar to be available to watch within the next couple of weeks. And finally, I would like to highlight that it's really more important than ever, as you can tell the pace of progress, to stay connected and engage with your fellow molecular bioscientists. So do join the Biochemical Society's community of researchers and specialists, and take advantage of the key benefits. This includes discounted registration fees for courses and meetings. There's exclusive access to a wide range of grants and bursaries, personal online access to 2 journals, and so much more. So do visit the website. And I think at this stage, after the most really innovative webinar, where I feel we've just all learned so much from Ivan, thank you so much. Thank you to everybody, and goodbye.
Thanks a lot.